Funding for this program is provided by Annenberg CPB to advance excellent teaching.
They called it the decade of the brain, the 1990s, a time of rapid advancement in the field of neurobiology.
Neuroscience as a field had matured to the point where the information was gathering at a very, very rapid pace.
I think the 1990s were a decade of incredible progress in neuroscience, but I think it's only the tip of the iceberg.
But the revolution in our understanding of neuronal function continues. Knowledge of the mechanisms at work at the molecular level, coupled with innovations in imaging. Researchers see and learn things they never would have guessed, and there's no telling where the field of neuroscience will go from here. Talk. That's pretty much what neurons do. Like operators in a 24-hour call center, neurons communicate constantly. But make no mistake about it, their conversations are complex. It's taken a long time to figure out what's being said and how to listen in. Research underway around the world examines neuronal communication and the molecular foundations of cell-to-cell signaling. Neuroscientists continue to uncover new ways to see the neuron in action. And the more they look, the more they can see how much remains to be learned. Wolfhard Almers is a senior scientist at the Wallem Institute at Oregon Health and Science University. He is interested in the molecular processes involved in the neuron's primary activity, cellular communication. What can study of the action of neurotransmission tell us about how these complex cells manage to function? And what does neurotransmission actually look like?
There are probably 1 billion neurons in our central nervous system and about 1 trillion connections. There are neurons that send a signal to only one other cell, and there are other neurons that get input from, on average, about 1,000. Also, it varies quite enormously. There are big neurons and small neurons. So neurons are cells that have two ends. The one end is called a dendrite, basically a tree that branches in many directions. Through a dendrite, a neuron can receive maybe anywhere from 10 to 10,000 inputs.
The other end of the neuron is the axon. At the ends of the presynaptic neuron, electrical signals trigger the release of neurotransmitters, which then bind to receptors on the postsynaptic neuron, continuing the signal.
So neurons can make many connections and will make many connections. So they both listen at the same time and talk at the same time, unlike people who have a hard time listening and talking at the same time. But neurons can do that.
The neuron's ability to listen while talking is facilitated by vesicles at the presynaptic terminal. Loaded with transmitters, they stand by, ready for release.
So a vesicle is like a soap bubble, very small, and it's surrounded by a membrane, just like a soap bubble is surrounded by a film of soap. And inside is the neurotransmitter.
Once an action potential has reached the terminal, the vesicle fuses with the presynaptic neuron's membrane, and the transmitter is released into the synapse.
And the outcome is that what was inside the vesicle, the transmitter, gets to end up outside the cell. That's called exocytosis, and that's how neurons release transmitter.
In studying the process of exocytosis, Almers wondered what it might look like. How do vesicles approach the membrane before fusing?
We were always interested in capturing single events of release by one method or another.
But before the 1990s, the only way to see live activity in real time was by measuring electrical charges in the cell.
There's one problem with electrical recording, and that is that all you can ever find out is when exocytosis or when the fusion event happens. What happens before, what the vesicle does before it fuses, you can't tell. But you can tell if you can see vesicles and watch them. And this is what we wanted to do.
Thanks to improvements in imaging technology devised during the decade of the brain, Almers got his wish: the ability to integrate robotics, light microscopy, and computer software enabled Almers to view vesicle activity more clearly. And in the year 2000, he caught it all on tape.
To see things happening in a live cell, you need to use light microscopy. As the price you pay for that is that it's harder to see very small things because the lenses that we use on microscopes, by the very deep principles of physics, blur small things so they look large. It's okay if you can't recognize its face. If you just see that something is there, that's already good enough if you want to tell whether it fuses with the plasma membrane or whether you want to see it approaching the membrane or wandering around in other ways. So you don't need to see its face. You just need to see that something is there.
And because Almers was able to attach a video camera to a high-powered light microscope, he can now show others what he saw.
We can see vesicles approaching the plasma membrane, and we are interested in studying the molecules that help it approach. We can test whether it's actually molecules that are needed to do that or whether the vesicles can just rattle around, you know? So information in a living cell as to how long it takes for an event to take place is very important. What we are interested in now is to actually see the molecules being recruited to where the vesicle sits.
Additionally, Almers knows that a deeper understanding of the mechanisms of neural transmission can help shed light on another recent discovery about neurons. Conventional wisdom has held that most neuronal connections form during the developmental years and don't change much over the course of a lifetime. But researchers have found that change actually occurs on a daily basis.
While we are speaking, and every morning you wake up, you're the same person almost. You're never quite the same person because through the day's experience, your synapses will have changed as a result of transmission. So neurons are wired together in a very complicated set of connections. And we are only beginning to understand how complicated it is and how it changes.
The idea that synapses are more plastic than fixed is supported by continued research and observation of how neurons behave. One area of study that forges ahead as scientists understand more about how neuronal connections change is the study of learning and memory. Researchers find that learning involves a lot of change at the molecular level, and by mapping out the molecular processes involved in the formation of memories, they have established connections between physical changes in the cell structure and the ability to remember.
Basically, the brain works by communication between neurons, and it's this continuing communication between neurons that's important for changes in synaptic connections that underlie things like learning and memory or any response to our environment.
In the lab of Richard Hugoneer at Johns Hopkins University, researchers examine memory at the molecular level. They focus on changes that occur at the synapse as new information is processed and committed to memory in the brain.
Synaptic plasticity is critical for learning. To actually process information and come up with something new, you really need to form new connections. What most neuroscientists believe is that to actually encode memories into our brain, you need to set up a network, a neuronal network, that can be recalled when you remember something. And so the idea is that there are millions of neurons, and that when you are learning something, you're setting up a new network through those neurons. You're enhancing certain connections between certain neurons and actually inhibiting connections between other neurons. And in this way, you scoped out a pathway, a neuronal pathway through this network of neurons. Many of us are interested in what are sort of the molecular mechanisms that underlie this process of both strengthening and weakening the synaptic connections. So at a molecular level, how do you make neurons communicate better?
To explore questions of neuronal communication and the importance of strong connections to the formation of memories, Hugoneer's lab focuses on the process of long-term potentiation, also known as LTP.
Long-term potentiation is a process whereby synaptic communication between neurons can be modified for long periods.
In normal neuronal communication, input from a presynaptic neuron leads to firing in a postsynaptic neuron. Increased stimulation from the presynaptic neuron causes increased firing of the postsynaptic neuron. But in LTP, the postsynaptic neuron continues to fire at an elevated rate, even after the increased stimulation has subsided.
So there's been a large effort to try to understand how such a brief, a few second stimulation can convert a synapse into a more potent synapse, and how this can last for a long period. So we've been trying to understand at a molecular level what occurs at the synapse.
There are two explanations for what actually occurs at the molecular level during LTP. One hypothesis suggests that the presynaptic neuron fires at a normal rate, releasing an elevated amount of neurotransmitter.
If you release more neurotransmitter, you're going to get a bigger response in the postsynaptic neuron. The other way is actually to, for example, modify the receptor function so the receptors are more sensitive to the neurotransmitter.
In this second model, a normal amount of neurotransmitter is released, but the increased sensitivity of the receptor causes the postsynaptic neuron to fire at an elevated rate. While it's clear significant changes occur on both sides of the synapse, Huguenier chooses to focus on the role of postsynaptic receptors for the neurotransmitter glutamate. He finds that LTP actually increases the sensitivity of these receptors, strengthening the neuronal connections that are formed as memories are made. When the neurotransmitter glutamate binds with the receptor, it opens up a channel through the membrane. Electrically charged ions enter the postsynaptic neuron, which then fires an action potential. But a heightened state of sensitivity occurs when an enzyme phosphorylates the protein channel. The next time a neurotransmitter binds with the receptor, more ions stream through the channel. More action potentials fire within the postsynaptic cell, passing the strong signal along to other neurons in the network.
It actually passes more ions per second through the ion channel. So for the same amount of neurotransmitter binding to the receptor, you actually get a bigger flux of ions across the membrane.
The lasting change in receptor sensitivity to glutamate is the first step in a complex web of signaling cascades that can lead to stronger neuronal connections. Genes are turned on that reinforce the structures at the farthest ends of the axons and dendrites, thus stabilizing them. Most researchers believe this synaptic stability is the molecular foundation of memory. Repeated exposure to certain stimuli may set off the chain reaction required to make a lasting impression. Researchers know there's still much work to be done before making direct connections between brains and behavior, but they appear to be well on their way to figuring it out.
We've come a long way, especially in the molecular analysis. Really, I think we're at the beginning of a whole new field of really how the brain works. Although we understand how synapses work to a certain degree, we have very little idea how higher brain processes occur. And I think probably that this is going to be the century of the brain, and we will really begin to understand how higher brain functions occur at a more what we call systems level of neuroscience, and possibly even things such as consciousness.
Researchers still have more questions than answers about all the mechanisms involved in neurotransmission and the formation of synaptic connections. But one way to understand effective communication between neurons is to observe and measure how they respond to different stimuli. Exogenous chemicals introduced into the system activate cells, enabling researchers to explore a range of neuronal responses. In the lab of John Williams at the Volum Institute in Portland, Oregon, researchers carefully observe the electrical behavior of cells that comprise a network known as the reward pathway.
The reward pathway, it's actually a pretty extensive pathway, but the heart and soul of the reward pathway is a group of cells in the midbrain called the ventral tegmental area.
Neurons in the ventral tegmental area send signals along the network to the nucleus accumbens, the prefrontal cortex, and the amygdala.
endogenous reward is really important for survival, right? People like to eat. Sex is very important. All the things that people enjoy work through this pathway. So it's a real important survival area for everyone, basically. It's called the reward pathway because animals will work to have stimuli in the ventral tegmental area. So the activation of these cells is thought to be rewarding because animals will do things to get those cells to be stimulated.
Neurotransmitters are the main agents of stimulation. These chemical messengers help the brain perform a range of functions, from cognition to emotion, depending upon their release and subsequent interaction with receptors on the postsynaptic membrane. Neurons are classified by the specific types of neurotransmitter they release: GABA, glutamate, and dopamine, to name a few. Along the reward pathway, the neurotransmitter dopamine dominates. Neurons that traffic in dopamine are most common here. They produce, package, and release the chemical messenger into the synapse.
If we're talking about dopamine as being the neurotransmitter, the receptor is called a dopamine receptor.
In the Williams lab, researchers examine how dopamine cells from the reward pathway respond to different chemical stimuli. They work to tease out the mechanisms of addiction by studying molecules that may alter the neuronal structure when exogenous chemicals are applied.
We study rat brains. We use the brains from animals that have developed normally. We take thin sections of the brain, we call them brain slices, and we can actually identify the cells that we're recording from, and we look at the excitability of the cells using recording electrodes.
Williams introduces a variety of substances to stimulate neurotransmitter release and test neuronal response to drugs of addiction.
We can also apply drugs exogenously so that we can really test the effects of the endogenous compounds, the neurotransmitters, as well as exogenously apply the neurotransmitters, as well as the drugs of abuse. All drugs of abuse seem to speed the activity of dopamine cells. There are not many drugs of abuse that directly activate dopamine cells. Example of one that is nicotine. Nicotine activates dopamine cells directly. Things like cocaine cause an inhibition of dopamine cells. So we'll take a normal animal and give them cocaine and see how does neurotransmission in these dopamine cells change. We'll study the adaptive mechanisms that happen after treating animals chronically with cocaine.
Adaptive mechanisms include lasting changes in cell structures. The principal method by which the Williams lab observes both short and long-term changes in dopamine cells is through the use of a technique called patch clamping.
Patch clamping is just a method to make recordings from cells. It involves taking a glass pipette and making a very tight seal between the glass and the neuron that we're studying. The glass pipette is like a little straw, so once you make the seal between the straw and the cell, then you can disrupt the membrane, the plasma membrane that's on the inside of the pipette, and have direct access to the inside of the cell. So now you've got very good electrical continuity between your pipette and the inside of the cell. So now when channels open or close in the cell, you're able to record the ion currents that pass through those ion channels. By recording the activity of cells, you can say, does this transmitter excite the cell or inhibit the cell? What does the drug of abuse do to the excitability of the cell? And then you can determine which ion channels are opened or closed by the neurotransmitter or the drug. You can also study the molecules that are controlling the channels inside the cell.
Williams hopes such close examination of dopamine cells from the reward pathway can uncover some of the mechanisms of addiction. How do drugs of abuse begin to control rewards?
So, like we talked about before, the reward pathway is very important. And what drugs of abuse do is just take over that pathway so that perhaps even without any kind of endogenous reward, the drug takes over that pathway, and that's the problem. It basically floods the reward system with no real reward happening. And then it makes it seem like the normally rewarding things are less rewarding. The hope is we'll be able to discover, by knowing these adaptive changes, be able to make some comments about what really happens during addiction.
Imaging techniques have radically improved our ability to look at brain cells. And the revolution in our understanding of the neuron continues as researchers explore ways to apply imaging technologies to the study of neuronal plasticity. Scientists can see cells grow and change. They never conceived of all the ways. At the end of the 20th century, researchers made one particularly surprising discovery: reservoirs of new neurons in adult mammalian brains.
There was a belief that once the process of development has ended, postnatally, presumably, there was no substantial amount of new neurons that were born into the brain. There was this general idea that the analogy for a brain's function is that of a computer. And it's a fixed process, and memories are stored in these circuits. And how could you possibly add new cells into the circuit and not disrupt them in some ways?
At the Salk Institute in La Jolla, California, Fred Gage has found the opposite is true. He works with new neurons, stem cells in the brain that grow and form specific connections well into adulthood.
And it really wasn't until the early 90s that technology had advanced far enough so that one could use, for example, confocal microscopy along with antibodies which would specifically stain for elements of neurons which were convincing. And then you could multiply label the cells.
The ability to label specific cells and look at them in 3 dimensions, as Gage does in his lab now, has given researchers the chance to observe their growth over time and see they are, in fact, functioning neurons.
Confocal microscopy was a really important element to this whole process because the allowed one to do was to optically dissect with a laser the tissue and take pictures, digital pictures, all the way through the cell and then reconstruct the cell in 3 dimensions to prove that the cell that had been labeled had, over time, differentiated into a fully mature cell.
This new technology allowed Gage and his team to identify 2 areas in the adult mammalian brain where cell proliferation clearly continues, in the hippocampus and in the subventricular zone.
Confocal microscopy really nailed it down that neurogenesis was occurring, and the significance of it was further amplified by what are called stereological quantitation. So these are methods that have been developed over the last 10 or so years that can accurately count how many cells of a certain type exist within the brain. It became clear that there actually were a lot of cells that were being born in the adult brain throughout life. It wasn't just a passing phenomenon. And it was also shown that they persisted throughout life. So they weren't just born and died off, they actually persisted.
Once satisfied that neurogenesis does in fact occur in adults, Gage turned his attention to possible factors in the growth and functionality of new neurons in the brains of mouse models. He explores possible connections between enriched environments and increased neurogenesis.
We had a surprise that occurred some 5 or 6 years ago where the events of neurogenesis were modified by changes in environment. If you take genetically identical, sex-matched, so the same gender, same age, adults, and then split them into 2 environments, one which is standard laboratory conditions, impoverished as you might call it, versus the littermates or the siblings, which are placed in an enriched environment with toys and much more space and things to interact for merely 45 days and then look at their brains. We found that there was as much as 15% more neurons in this area of the hippocampus as compared to the control. We increased the number of cells that survived and integrated into the circuitry. Part of our effort now is to try to unravel these correlations to see what is it about environmental stimulation that causes this increase in neurogenesis. So as a neurobiologist, I believe that the brain is the controlling organ for our behavior. But now we're finding out that behavior can change the structure of the brain. So what this means is that who we are as a function of our brain is affected by what we do. And it's a bit of a do loop, if you will, where the brain controls behavior, but behavior then can feed back and regulate how the brain then will come and respond to that next environment.
Beyond understanding possible links between behavior and brain cells, Gage hopes the study of neurogenesis might lead to new kinds of therapies.
If we could learn enough about molecules that are involved in migration and proliferation, we might be able to put up together certain kinds of strategies that would take advantage of this residual plasticity. I'm very optimistic in the near future that there will be strategies for inducing what we call self-repair within the central nervous system by taking advantage of these endogenous cells that exist.
Researchers continue to eavesdrop on cellular conversations, trying to figure out what's being said, how signals are transmitted, and what can be done to facilitate the process. The more they learn about neurons and all their complexity, the more they find there is to learn about them, and how the sum total of cells, synapses, and chemical messages all come together to communicate and change through the course of our entire lives.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.